for the invitation. It's a pleasure to be here. I was just talking to Bazak. It's been wonderful to have these presentations in general. Every month there's like this awesome meeting with awesome questions. So hopefully this will keep up. I know it's a lot of work for the organizers, but thanks again for organizing this. So as Bazak mentioned, the name of my talk is Correlates of Temporal Processing in Humans. And what I intend to do is show some of the stuff we've been studying in the lab, at least some of the studies that we've been focusing more in using EG to understand how we process time. So of course, for this crowd, I don't need to explain how important time is and how it underlies a lot of our interaction with our environment. There's a lot of studies that have shown that. And our interest, as I imagine it is the same interest as many of the crowd here, is to understand how we track time. So how do we keep track of time? And has been different models. This is just a small choice of some of them that Virginie presented in a chapter a couple years ago. So you see that you have all different sorts of models. Some will say that, you know, to keep track of time, we have these intrinsic networks that time can be pretty much computed in any neural network. We have other models that we propose that we have actually some kind of central dedicated mechanism that tracks time. And we have different variations of these different models. And again, one of the questions is, although the time literature has done a great job in proposing on these models, one of the questions is, why do we have so many models? And how can we start choosing between models, or at least understanding which model fits better to different tasks? So one of the answers is, you know, why do we have so many models? One of the answers is because different models were proposed to explain different findings. So we have this whole set of models that are really interested in very short time intervals, and then the intrinsic models work quite well. You have other models that will try to explain different aspects of temporal processing. So some models are more focused in explaining rhythmic processing. Some models are more focused in explaining how we can use time to track longer intervals. Some of them will try to say, how can we use time to anticipate events? So we have all these different models that try to explain some findings. There's some overlap between them, but we have different models that will try to explain very specific findings. And then, of course, we also have very different levels of description. So you have some models that will try to make a very psychological model, very high level, and some models will try to go down to the neural level and saying, well, this is how this should be implemented in the brain. And again, different models will have different focus on this with different objectives. But then again, there's another problem, is that we have also an overlapping behavior predictions. So even models that work with similar tasks and try to predict similar durations, sometimes their predictions are quite similar to each other. So sometimes just looking at behavior does not help to associate or discriminate which model is better. And this is true within each of the small groups of models that will try to explain one temporal task or one temporal ability or the other. And how can we overcome this? So again, part of this is not really a problem, right? So it's okay to have different levels of description. It's okay to have different models that will try to explain different behavior findings. But there is the problem of having at least some models that will have this overlap in predictions. So how can we try to overcome this? One way would be to look at some kind of brain imaging method. So if you have models that have an overlap in behavior predictions, what you could do is try to see how the brain is reacting in these different tasks. So you could look at EEG measures or fMRI measures and try to understand which of these models predict better, or at least is more makes better predictions of what would happen if we look at EEG. Of course, this is not new, but the problem is, for this to work, we would need clear electrophysiological correlates of temporal processing. So to be able to say, the brain is processing time this way or that way, it would be important to know what we're looking at or what kind of EEG signal we're looking at when we're processing time. And similarly to models, we also do not have yet a clear EEG correlate of temporal processing. We have several proposals. So this is a chapter written by Tadeusz, Hedrick, and Mac a couple years ago. And you see that you have different papers that will propose different correlates of temporal processing. Not only different correlates, but different moments where you should look for those correlates. So some people would say that, some people found that beta power before the target, the interval starts, is correlated with temporal processing. There's a lot of studies doing the interval to be timed, and then the CNV is the most famous one. So you have this component that seems to correlate with some aspects of temporal processing. And recently you have this increase in studies that will look at post-interval periods. So they would try to look for neurosignatures of temporal processing at the offset of the interval. So when the interval to be encoded or to be judged or to be reproduced ends. You have different studies that have looked at this. So one, Tadeusz did one study in 2015, I think, where he found this N1P2 effect. There are some studies that find the P3 and the LPCT. The last meeting, Mir, who is working with Ayat Landau, also showed some kind of P3, LPC kind of response. So there's this increase in studies that will try to look at this post-interval period. And again, why do we have so many signatures? Again, because one of the problems that within a given task, it's not that easy to associate what those neurosignatures are actually correlated to. So within one single task, it's not easy to know whether that correlates, be it beta power, alpha power, the CNV, some kind of post-interval activity, if that's really something that's tracking time in general, or if that's somehow tracking some kind of task-specific demand. To be able to have a better idea of what would be neurosignatures of time, ideally for each time we would have a perfect control. But then to have good controls for temporal tasks, it's really hard. First, because one normal confound is you always need to have something that kind of is similar in difficulty. So ideally you would have some kind of task that does not involve time, but is as hard as the task that you're looking at. Ideally, you would also have a task that would control perfectly for any kind of stimulus features. And finally, you would need to find something that changes in one direction at a fixed pace, which is pretty much what time does. So in a lot of these tasks, when you're trying to track time, what you're doing is you're kind of integrating some kind of magnitude that only changes in one direction, can only increase and at a fixed pace. And anything that you do that will perfectly correlate with that will, by definition, also correlate with time. Again, there's a lot of interesting ideas of how to tackle this. So a couple of meeting ago, Froch presented some of his results with numerosity. So number seems to be a nice way to kind of try to find a way around this. But again, this is just to show how difficult it is to find good controls for time tasks. So what has been our approach? So what we have done is to look at EEG activity across different tasks and temporal manipulations. So given that there's no one perfect control, what we've been trying to do is just do different tasks that have some overlap with each other and just deal that each of them has its own limitations. But in the very long run, we can try to put all these results together, and hopefully these results will help us to limit and discriminate which models are better. So although each task by itself won't be perfect, putting them all together, we can see what comes up and what survives or what normally correlates with timing in these tasks. So this is mainly what I'm going to present. So here I'm going to present 4 studies that we did in the last years. And the general objectives of these studies were to compare different patterns of EG activity across different tasks and temporal manipulations. And we had two main questions. One of them was, what patterns of EG activity are really task specific? So what kind of patterns only emerge when people are performing a time task? Or what patterns emerge when they're performing one specific time task in contrast to another? And then in turns 3 and 4, I'm going to focus on what kind of EG activity is correlated to the duration, to the actual duration of an interval, or to some other decisional aspects of the task. Okay, so this is the first experiment. So this is a study. I presented part of it some years ago in Mexico. It was published last year. And in this task, what we did was we did a temporal and color discrimination task. So this study was performed by Fernando, who is a PhD candidate in the lab. And it was based on some studies by Jennifer Kuhl that already investigated this relationship between time and color. So it's a quite simple task. What happens is in each trial, participants will see a first target, and then after a while, they will see a second target. This first target has a given color and a given duration. So the duration could go from 700 to 1500 milliseconds. And then the second target would have a different color and a different duration. In different blocks, participants would have to judge either the color of S2 relative to S1. So if the second target was more red or more blue than the first one, or whether it was longer or shorter than the first one. So this was performed in different blocks. Critically, in some blocks, participants knew beforehand what they were going to judge. So in some blocks, in all trials, they had to judge color, for example. In other blocks, in all trials, they had to judge time. And then in some blocks, they would not tell them in advance. So they would see the two events, and then afterwards, we would ask them, okay, so now judge whether the second target was longer or shorter than S1, or whether it was more red or more blue than S1. So this is quite simple, and again, we can look at two main aspects of this task. We can look what happens in EG activity when participants are encoding the first event, and what happens when they are making a decision, when they're looking at the second event and making their decision. So first of all, when we look at behavior, we can see that participants can perform the task quite well. So this is just showing that as the second interval gets longer, participants answer appropriately, saying that S2 was longer than S1. Also for color, and also knowing beforehand whether they will have to judge time or color also improved performance. So they did better the task if they knew beforehand whether they would have to judge in that trial time or color. Okay, so then looking at the actual activity, so we're going to start looking at activity of S1. So remember that in this part, the participants are seeing the screen is pretty much identical irrespective of the task. So they're seeing an event that has a given color with a given duration. And the question is, they knowing or not what they're going to have to do with that event if that would change EG activity. So to look at that, what we did was we used a multivariate distance. So I won't get too much into details, but this is mainly showing how different the EG pattern is for each time point across all sensors. And this is just comparing tasks pairwise. So for example, the pink line here is, you can see my mouse, right? You can see my pointer, right? Okay, so the pink line here is just showing how different the activity was when participants know that they're going to have to use this information to make a temporal judgment or a color judgment. And to our surprise, we didn't have that much of a difference between tasks. So during the encoding, if participants knew they would have to make a time judgment or a duration color judgment, we didn't really change the pattern of activity. We can also look at the S1 offset. So because these events have different durations, we can also look what happens when the duration ends. And again, when we look at this, there's no clear pattern of differences. So there might be some difference here. So the green line is actually time per times compared to color peer. So there seems to be some difference, but it's not that consistent and it doesn't reach significance. So it does change a little bit across participants. So this is showing that at least during the encoding of the information, knowing that you're going to use that information for time or for color doesn't really change the pattern of activity, at least the activity that we can capture with EEG. What about in the second event? So now participants already saw the first event, and now the second event comes up. And for example, if they have to judge color, they know that that's where they have to compare it relative to the first event. If they have to judge time, they know now that they have to pay attention to time because that's the relevant feature that they're going to have to compare afterwards. And what you see that in this case, there's a huge difference between tasks. So the green line here is showing how different the activity is when you look at the onset of the second event, when participants have to judge time or color, and you see there's this huge difference. When they don't know what they're going to judge yet, there's no difference, which is expected. And if they kind of know what they're going to judge, there's this mixed pattern of differences. And this is true for both the onset of S2 as for the offset. And this is important because remember that when they're going to judge time, at least when the second interval is shorter than the first one, you really need to wait until the offset of the second event for you to be able to make your decision. And again, here, the offset, you can really see that the activity evoked by the offset of the interval is really different depending if whether you're going to use that information to judge time or to judge color afterwards. So this is just showing a combined pattern across all sensors. So where is this difference coming from? So this is one way of seeing it. So this is just some topographies at S2 onsets. And what you can see here, I'll start with the second row, which is color pure. So this is when the second event comes up, and you see that after 300 milliseconds, you have this huge P300, which is pretty much people making their decision. So now they have enough information, they can judge, this target is more red or more blue than the first one. If participants are tracking time, you don't really see this P300 well defined. And again, if they might track time or color they don't know yet, then you see really this kind of mixed pattern between these two patterns. So you see this P300, it's not as big as when they know they're going to judge color, but it's still there. Again, here, for the color decisions, you can make up your mind as soon as the second event comes up. But for time, in several tries, you're going to have to wait until the second event ends. So if we look at this kind of topographies for the offset, you also see this interesting pattern. So for time pure, what you see is that when the second event offsets, you also see this pattern of activity around 200 to 400 milliseconds, kind of these central posterior electrodes, which is not present in color pure trials. So here participants, they already know what they're going to respond probably. They already made their mind. And again, in time mix and color mix, you kind of see this mixed pattern. So this is showing that this pattern of activity, this kind of EEG signals only comes up when participants will have to make a temporal decision for that event. And this is probably where all these differences come from. And then, of course, the second question is, so you do have this different activity that seems to be really dependent on whether people are making time judgment. But does this activity also correlate with the duration of the events? So is it just an activity that shows that this is a time judgment or some kind of signal that correlates with how much longer or shorter that interval is relative to the first one? And what we see is that it does correlate. So we have this, so this is the offset. This is showing how much the EEG pattern correlates with time. And then when you look at each task independently, you can see that for the time pure, so this is really the task where people know they're going to have to make a judgment about time. You see that after it's offset, you have this really nice and clear frontal central signal that correlates negatively with time. So again, this is really similar to what we presented last week, and you can see that similar to what he found, you have the signal that it's more present or it's stronger when the second event is shorter than the reference. When it's longer, which is these lines in the bottom, you don't really see that difference. Interestingly, this also only shows up if people have to make a judgment about time. When you look at the same signal for color, you see pretty much everything's kind of flat here. There might be some kind of drifts, but in general, this really clear pattern is only present when people are looking at paying attention to the duration of that second event. And if they might have to make a judgment about time, then the activity is kind of there. It's not as pretty as when they know they're going to have to make a judgment about time, but it's more present than when they don't know, when they know they're not going to have to make a judgment about time. So this really shows that this activity is really time dependent. It's only present if you have to make judgments about time, or at least about some kind of temporal related aspect of that target. We also see an early activity, which is kind of like a P300, N200. It's more occipital. It's not as clear, but you also have some kind of early activity that seems to also correlate with this. When you look at that, when you look now just at these two activities, again, you can see this pattern. So this is the late frontal central activity, and you can see this nice monotonic pattern, just as Neil presented last meeting. And you can see that this is really strong when time is a relevant feature, kind of there, but not as strong when time might be a relevant feature, and pretty much not there when time is not important at all. And for the first activity, the early one, you kind of have the pattern where almost independent of whether time is relevant or not for the task. Okay, so in this first experiment, what we saw is that during the encodings, when participants were looking at the first event, we did not find strong differences in EG activity. In the second event, we did find strong differences, and there was two main patterns. First was this kind of late P300, late positive component activity that was present mainly when participants are making temporal judgments. And this early P200 activity that in this task did not really depend on that task. It seemed to be there almost all the time. Okay, so afterwards we perform another experiment that's similar to the first one. So again, we have two events, but now what we wanted to look at was whether there was a difference in EG activity within timing tasks. So again, in both cases, you had a first event. Here, instead of making a continuous duration, the first event could last for a possible duration, so from 750 milliseconds up to 2000 milliseconds or 2 seconds. And then what would happen is that in some blocks, participants would have to see this first event, then a second event would come up, and they would have to judge whether the second event was longer or shorter than the first one. And in other blocks, they would see the first event, and they would have to reproduce it. So there was mainly a difference between encoding time to make a temporal discrimination task or encoding time to make a temporal reproduction task. Here, participants always knew beforehand the task they would have to perform. So it was either a full block of temporal discrimination and then a block of reproduction and then another block of discrimination, but they always knew beforehand whether they would have to make a temporal judgment or a temporal reproduction task. And what we see first in the behavior results that participants can perform the task quite well. So they can easily judge whether the second event was longer or shorter than the first one. And the reproduction task, they can also reproduce these regressions quite well. You see the classic regression to the mean effect. So this is just showing that this works quite well. And again, our first question was looking at this first event, and for this experiment, I'll focus mainly in S1, because this is where really both tasks are identical to each other, whether we can decode whether participants are going to use that information for some kind of temporal discrimination or reproduction. And what we see is, looking at the onset of these activities, that you can actually, there's a marked difference in activity when participants are encoding information to either perform a temporal discrimination task or a reproduction task. So you can see that quite early, in the duration of that event, but more consistently a little bit later, there's a difference in the EG activity across our sensors when you look at whether that participant is going to perform a reproduction task or a discrimination task. When we try to see where this difference is coming from, so this is just showing some ERPs from different regions, so frontal, temporal, central parietal, and occipital, you see that this difference is mainly in these frontal central electrodes. So when participants need to perform a reproduction task, doing the encodings, remember they're not doing the reproduction here yet, they're just learning the interval they will have to reproduce. You see this more negative activity in frontal electrodes than when they have to do a discrimination task. We can also look whether this activity is also correlated with the duration of the event. So remember that we have 4 durations. So we can look at the final milliseconds of each duration and see whether this activity for each of these durations is different enough from each other that we can decode time. And what we see is that for temporal reproduction, we can actually decode time quite well. For discrimination, not that well. So again, what we're looking here is looking at the last 100 milliseconds of activity of S1. Nothing's on the screen yet. People just pay attention to the interval that they might have to either reproduce or judge afterwards. For the reproduction task, look at this activity, or this activity is correlated with the duration of that interval. And for discrimination, it's not that strongly correlated. So again, this is interesting because it's different than what we saw before. So here, yes, we can decode whether participants are going to use that information to reproduce or to discriminate an interval. And especially for reproduction, apparently this activity also correlates with how long that given interval was. Again, here we're looking at S1 onset. So we're just looking at when the first event started and has not yet finished. But we can also look at S1 offset, so at the end of that interval. And here, again, what's interesting is that we can actually decode both sources of information. So we can decode the task participants are performing. So if they are doing a temporal reproduction task or a temporal discrimination task, but we can also decode the duration of that interval. So both sources of information are present in the offset signal. And again, we can check whether we can decode time in both tasks, and if there's a difference in how well we can decode time across tasks. And what we can see is that we can decode time in both tasks, but apparently, again, for the reproduction task, we can decode time a little bit better. And if we want to see where this decoding comes from, we can look at the progress of this effect, and you see that this decoding kind of follows the same pattern that, so this is the regression coefficients for the regression, and you can see that this coefficient starts quite in these early sensory areas, and then they kind of go to more late central areas. So what this is showing you is that when looking at the offset of the activity, we can first decode whether they're going to use that information for a temporal reproduction task or for a discrimination task, and the actual duration of that interval. And it seems that we can decode better when they're going to use that information to reproduce a target and not to discriminate it. So here we can find differences in EG activity when participants are encoding time for different timing tasks. Of course, this got our attention. So in our mind, or at least in my mind, I thought a timing task versus a color task should be more different across each other than 2 timing tasks. But what seems to be happening here is that the reproduction task is really different than the others. So it seems that these differences are being driven mainly by the reproduction task. And time-related information could be decoded better in reproduction tasks. And where this information is in EG activity has a similar pattern that we saw before. So it starts in these early sensory areas and seems to migrate afterwards to this late decisional EG activity. Okay, so these 2 first experiments The question was how task-specific these EEG activities are. And the second two experiments, the question is these patterns of EEG activity, are these patterns of EEG activities correlated to the actual duration of the interval or some other kind of decisional aspect of the task? So we did two different tasks, two different studies to investigate this. The first one was by Mateus, who was also a PhD candidate at the lab. So we just submitted this to a preprint yesterday, so it might come out today or later this week. And this is a temporal discrimination task, so it's a little bit different than what we were doing before. So in this task, participants only see one target. So there's not S1 and S2. We go straight to the decisional phase. They already see the target they're going to have to make a judgment about. And in this task, the target could last between 500 to, so half a second to 2 seconds. And participant would have to judge whether this duration was longer or shorter than the reference. Now, the trick to this task was that we never told the participant what was the reference. What they knew was that in each block there would be a new reference, which they did not know what it was, and they would have to learn this reference by feedback. So in the first trial, they would see a duration, they would say, I think this is longer than the reference. They would get a feedback, and they would do this a couple of times, and eventually they would learn what is the reference they have to judge durations against. Participants did not know, but there was actually only 3 possible references, so either 650 milliseconds, 1100 milliseconds, or around 1.5 seconds. And they had to learn again these references just by their feedback. The blocks were quite short, so we were able to have a lot of blocks, because participants, they learned this quite quickly. I'll show the behavior responses soon. But you see that they can learn this quite quickly. So we had a bunch of blocks with these different references. And then, in the end of the block, we would ask them to show us what reference they were using. So we would ask them to reproduce the reference interval they were using to make their decisions. So this is the behavior. So again, if we want to see how these behaviors change as a function of learning, what we're doing here, we're splitting performance within each block in 2 windows. So the first half of the block and the second half of the block. And you can see here that even within the first half of the block, participants can already discriminate quite well the hidden reference. So they already learn, they kind of know already what they have to respond. But then towards the end of the block, this difference is even stronger. So this is just to show that participants, they can do the test quite well and they learn really fast. So even with a couple of trials, they can kind of already capture what is the reference they are comparing each interval to. This is just the same information, but now actually looking at what's a measure of what the reference they're using, and you can see that as the block progresses, these references are getting further and further apart from each other. And in the end, when we ask them to reproduce what reference they were using, you see that they do it quite well. So they can really kind of find out what they have to do in the task. So this is interesting because it allows us to look at the activity now and see what changes just because they're updating their reference. And this is what we have. So this is just a big picture of the EEG patterns that we have. So again, you see that we have this early occipital activity that seems to be correlated with time, and again, this kind of late frontal central activity also correlated with time. Again, this early activity, so this is the line plot for the early activity for the different references. You see that for the short reference, the activity is way more negative. The other ones is not as well defined. But what's interesting here is that when you look at the late frontal central activity, you really see this pattern once again, just as the first experiments of Fernanda and what we presented last meeting, was this pattern of the shorter the interval is, the higher this activity. But you see that it also depends on the actual reference. So for example, if you get this duration here, which is 1.1, for this reference, this duration is longer than the reference. in this case, it's the actual reference. In this case, it's shorter than the reference. And you can really see this activity kind of going up, and just changing and showing that this is not being driven only by the actual duration. It's really capturing somehow whether participants are going to judge if that's shorter or longer than the reference. This is just another way of showing these results. Again, this is the early activity. This is the late one. And again, here you can really see that, especially for this intermediate durations, you can really see this difference of amplitude by the reference. This is interesting for different reasons. So first, you can see that for really short or really long durations, you don't really see that difference. And this is interesting because this is showing that participants might not be, or at least this activity might not be really encoding how far away from the reference that interval is. Because remember that the 500 millisecond duration is quite close to the short reference, but really, really shorter than the longer one. But still, you see that the pattern does not change as much here. It does change, however, for those durations that change whether they're longer or shorter than the hidden reference. And again, we can now see how this changes as a function of learning. So we can look at this activity in the first half of each block where participants are still learning the reference, and in the second half where they already learned this quite well. And again, you can really see this pattern changing. So again, this is that duration that in the short block is longer than the reference. And you can see that in the first half, it's you still have some kind of amplitude here, and then it just vanishes away. And in the long duration, where this is shorter, you can also see this kind of coming up. So you can really see that this pattern is changing as participants are learning what the reference is, showing that whatever this is encoding both the duration of the interval, but also whether that specific interval is going to be judged afterwards as shorter or longer than the reference participants are using. Okay, so again, we see this P300 late positive component that is present when participants are making a judgment. Again, we see the same pattern of this, some kind of sign response, coding how much shorter the interval is relative to the hidden reference. And it really seems to code both the duration of the interval and some kind of decisional criteria participants are using. And finally, in our last experiments, we wanted to investigate these components a little bit more in detail. And one problem we have with the majority of temporal experiments is that there's always a strong confound between the duration of a given interval and its decisional context. So just use our own experiment as an example. In our experiment, the 2-second duration was always longer than the reference, independent of which block they were. The 500 millisecond duration was always shorter than the reference, independent of which block they were in. So because of these correlations, at least in some levels of the duration and decision, it might be hard to associate which signals are encoding which aspects of the task. So what we did was we designed a new task where we had intervals that its duration and whether it was longer or shorter than the reference were perfectly uncorrelated. So again, this is like the simplest task that we did. So in each block, participants would see the reference 3 times, and then they would be presented with 12 durations, and after each duration, they would have to judge whether that duration was longer or shorter than the reference. Now the thing here is that we had 5 possible references. So it could either be 0.35, 0.5, 0.71, 1, or 1.41. So in each block, in different blocks, participants will learn these different references. And the idea here is that if we look at these 3 central intervals, its duration and whether it's longer than the reference, it is the reference itself or shorter than the reference, is fully orthogonal. So we can really now look at which patterns of interactivity are more strongly correlated with the actual duration of the interval, or whether it's longer or shorter than the reference. So behavior the participants can perform the task quite well. It's not that hard of a task. And here we're looking at the F values for all sensors, all time points, for duration, and for category. So again, here we can really see what's the difference in EEG activity, for example, for a 500 millisecond interval, if that 500 millisecond interval is shorter than its reference, the actual reference, or longer than the reference. And we're looking here at the offset of the interval, which is where we found most of the effects before. And you can see that there is some information about duration, but there's a very, very strong information about the category of that given interval. And again, where do we see that? We see two patterns. This early activity, again, in this kind of posterior sensors, and again, this late frontal central activity, which seems to be really be capturing whether that interval is longer or shorter than its reference. We can take a closer look at this, and this is what we have here. So this is the early activity, and here you can see evoked potentials for each duration and each category. So this is when the target was, all of these ERPs are when the target has 500 milliseconds of duration. This first line is when it's actually longer than the reference. The red line when it's the actual reference, and the purple line when it's shorter than the reference. And you can really see this nice pattern, which does change as the duration of the interval changes, but this pattern of more positive as a function of more, as of a function of longer than the reference maintains irrespective of the duration. And when we look at this frontal central activity, we see the same thing. So again, for different durations, you can still see this kind of P300. And again, with the same pattern that we saw before. So the shorter it is relative to reference, the higher this amplitude. And you can see these both signals really showing quite clearly, surprisingly clearly at least when I first saw this, whether this interval is longer than its reference, or longer, shorter or longer. And again, the same pattern that will be seen repeatedly, this very consistent late frontal central activity, and this early activity, which seems to change a little bit depending on the task. Okay, so here, again, we find this similar pattern of these N200, P300 RPC responses. In this case, both of them seem to correlate really strongly with decisional aspects of the task. But I didn't show here in details, but we can also find information about the actual duration of the interval in the EEG signal. So it seems that both the actual duration of the interval and whether that interval is longer or shorter than its reference is present in the EEG activity. So what do we learn from all these experiments? Hopefully different things. First, in the first two experiments that we see that this effect seems to be stronger in the decision phase than in the encoding phase. So apparently when I encode this information, although there was a difference in the second experiment, it seems that the strong difference is always in the decision phase. We find this more robust findings in this interval offset. So during the first experiment, the second experiment, we were able to find some activity during the interval to be time. It seems that at the offset, we have a more robust pattern. And why? There are different reasons, but one idea is that maybe the offset of the interval is actually working with some kind of pinging mechanism. So it's kind of helping this different hidden states being shown in the EEG activity. In general, we do find this early activity, but again, even in our results, where this early activity is changes. So we found the first experiment in the speech 200, then we found it kind of like an N200 related to temporal processing. As I mentioned, Tadeusz found it in a N1P2 activity. Some people have found in different early activity as well. So For this early activity, the general pattern is it's normally there. It does correlate with time, but it kind of depends where you're going to find it. On the other hand, this late component seems to be more consistent across tasks and experiments. And in a way, this is consistent at least with these hybrid models of timing, where these local task areas will interact with the higher central timing system. So maybe that's why we always find some kind of early activity which kind of changes where it is. And it might depend whether it's vision, audition, if it's a target onset or offset, but it's normally there somehow. But then it seems that everything migrates to this higher central system, which seems to reflect in this kind of P300 LPC activity. I know my time is done. It's my last slide, sorry. So what about this late activity? So can this be related to decisional models of time? So one of the most influent models we have at the moment is the DGM, or the top DGM, proposed by Fuachi and Patrick. And can the LPC or this P300 somehow be related to this? It's tough to answer right now. In principle, it could. So in decision making, in the decision making literature, the P300 has been consistently shown to be correlated with aspects of the drift diffusion model. So some people have proposed that the P300 is kind of capturing the drift diffusion. Other studies have proposed that the N200 can mark the onset of the drift process. So these ERPs could somehow be related to these parameters. However, translating how these potentials are related to timing might be a little bit harder than thought. Now, one of the reasons is, especially when you look at the P300 literature, most of the models would assume that when you have easier decisions, you would have the same P300, but the latency would change. So you would ramp up faster. And this is not really what we see in general. So we really see a difference in amplitude. There might be some way to make sense out of this, but it's still not as straightforward as I would think. And this is it. So thank you again. Sorry if I spoke too fast. Hopefully people understood what I was talking about. And this is the lab. And thanks once again for the invitation.
Thank you so much for a very interesting talk. This was a beautiful set of experiments, really. So yeah, we have about like 10 to 15 minutes for questions. So if you do have any questions, please raise your Zoom hand and wait for your turn, or put your question in the chat and I can read it for you.
Hello, sorry.
Okay, so Jennifer, cool. You could go ahead.
Hi, Andre. Thanks for that talk. It was really great. So this is kind of an open-ended question. It's not really a very technical question. It's more a sort of what do you think kind of question. So the fact that you see these differences mostly at the decision stage rather than at the encoding stage, does that reflect something about the EEG methodology, that the types of measures that we're taking with the EEG are just not reflecting the encoding phase, the fact of just timing for timing's sake? Or does it reflect something sort of deeper, more general, the fact that maybe it's just not useful to time just for timing's sake? Maybe we only ever time in order to compare it to something else. So I guess it's a question about, is this a limitation of the methodology, or does it reflect something actually significant about timing?
That's a great question. And I don't know, to be fair, it used to be really straightforward. So it could be both things, right? So we know that EEG is blind to different sources of activity. So, I mean, if it's not in the cortex, we're not going to see it. And even if it is in the cortex, depending on what activity is, we're just not going to see it. So it might just be a problem with the method. But as you said yourself, it could also be something more interesting. So it could also be that, timing kind of comes for free. So you don't really you don't need to pay that much attention. If you're just looking at a target, you kind of already have an idea of the duration of that event. Again, another way of thinking, so it could also be a mix of these two things, right? So it could be there, and it just could be this hidden activity that EEG does not capture. And this is why we think that maybe when we look at the offset, and we're into this to the whole to Mark Stokes' proposal of this hidden states that we could see with pinging, that could be one way to see it. We did try to think about ways to measure that more directly. So maybe we thought about maybe we can just ping during the whole duration. So let's just show it an interval. Let's just ping several times and see if something comes up with that. But that kind of makes the task harder, right? So if people are trying to make a judgment about time and you have something flickering all the time, we know that changes time perception as well. So sorry, it's not probably the answer you wanted to hear, but it's just it's probably both things. I fully agree. And it would be nice to know which one of these it would be, right?
Yeah, I mean, I didn't really expect an answer because it's a difficult question, but I just think it's something interesting for us all to consider, really. And just you mentioned the fact that you see the effects mostly at the offset. The other thought I had was that maybe the fact that we see these effects mostly at stimulus offset rather than onset really reflects the fact that, you know, our perception of time is a reconstruction of something that's happened already. It's not something that we're actively doing all the time. The only way that we can perceive time is to reconstruct what's happened already, and so maybe that's why the offset effects are so much stronger.
Yeah, and again, so I agree, and that's something really difficult to control, because for most of our perception, that's not true, right? So when you see color, it's just there. But time does have this aspect that when it ends, that you kind of reconstruct this. So yeah, it's one of those things that it's really hard again to have some kind of proper control in a task. Maybe
if you'd used like a dynamic color stimulus, we might have seen something similar in the color task. Okay, I'll stop now because I've asked lots of questions.
So we have Martin Wiener.
Hi, Andre. Can you hear me?
Hello,
okay. Yeah. I'm going to try and make this question brief. Great talk. I have lots of specific questions, but I'm going to try and ask one also open-ended question. You can decode better in the reproduction than discrimination, right? That's what you were showing in that one preprint. Can you speculate on why? Like, why is reproduction better at decoding than discrimination? I have some ideas, but I'd like to hear what yours are. Thanks.
So thanks for the question. We have some ideas. I think so. Again, it might be one of those things that have a really boring explanation or a really interesting one. So I think the most boring explanation that we could find is that maybe when people have to reproduce an interval, they have to pay more attention to it. So it's a more fine-grained task, you know, so maybe they really have to pay more attention, and maybe that's somehow showing that, and that reflects in the EEG activity. We would have to make proper controls for that. We don't think that's happening because, you know, we have reasons to believe that people pay attention all the time, but that would be, I think, the most boring explanation. The other one would be that maybe, you know, maybe time already interacts with these motor areas and you're just making it stronger, right? So there's all these proposals that time and motor areas are tightly correlated. And maybe when people had to actually code this for to reproduce an interval, it's just stronger, it just works better. Or maybe that's the natural way of doing it. Or again, maybe, and I think that comes back to Jennifer's question, maybe when we encode time for discrimination, it's just in some kind of activity that EEG is blind to. And maybe the motor activity is not. So we've been trying to find some kind of explanation for that. The first control that we did do, so I didn't show it here, but this activity, it's different than the activity than when participants are actually reproducing the interval. So we were worried people were just kind of practicing already during coding. But when we look at the activity, it doesn't seem to be the same thing. It's more kind of this lateralized to the right side in the encoding. So again, it's one of these questions. It could either be that you're just encoding it differently in the discrimination task, and this differently is something that EG cannot see, um, or maybe that we do encode better when we have some kind of action, and I think you have some results that show that that is the case, right? So that would be an interesting possibility.
Cool, thanks.
Thanks.
Sophie?
Hi, Andre. Thanks for this very interesting talk. Can you hear me?
I can hear you very, very low, sorry.
Now?
Yes.
Okay, I'll try to speak loud. So thanks for this very interesting talk. I had a question about the first study. So if I remember correctly, you found these very nice graded offset responses to the second stimulus, especially in the duration task, or actually only in the duration task. And it reminded me of the implicit timing literature. So the 4-period paradigms, where many studies have shown that you also see these timing responses when time is not the feature to be judged. So as in the timing comes for free, as you said before. So I don't know if I missed something in your paradigm that would explain why you don't see anything in the color judgment task, or I don't know, maybe you can comment on this aspect.
Yeah, so mainly, so you're right. So we did see only in the explicit, in the timing task. We didn't really see it in the color task. It is a good point. What I think might be happening is that even the 4-period task, although it is implicit, you do use time, right? So even though your task is not to judge time, you can still benefit from time to make your response faster. And here in the color task, time was totally useless, right? So in the color judgment task, well, all tasks were unspeeded responses. So maybe people they didn't use time whatsoever. Maybe if we had some kind of speeded response, we could also see this pattern emerging for tasks where time was not explicitly used. And this is something that we've been thinking about doing as well. So again, maybe it was, well, maybe in the 4-period task, again, maybe the thing is you're using time, at least some way, it's implicitly, but maybe then in that case, it would also be there. Just before, so in the last part of Jennifer's question, she said, well, maybe you could have used a dynamic color task, and this is something related to this. So one reason that we avoided that was that in this kind of task, you could also use time somehow, right? So maybe if it was a dynamic color task, people could also kind of find out whether it's more blue or more red, averaging for how long each color was in the screen. So maybe if you started using tasks where people can benefit from time, even implicitly, you would kind of see this LPC back. And this is where our first task was really, I think it was quite radical in that sense. Like in the color task, the time was not useful for any kind of aspect of the task. I don't know if that answered the question, sorry.
Yes, it does. Thank you.
Okay, thanks.
Finally, Pat.
Thank you, Andre. That was great. I'm just trying to understand. So basically, what you're seeing is a signature of a decision process. It's different for time versus color, but it doesn't seem to be any kind of indication of the mechanism by which you keep track of time. So it's interesting. Is that correct so far?
I guess so, yeah. So it's only the last part. So it is different for time and color, but so why did you say that it's not?
Well, I mean, if it was just, if you think about color and time as feeding into some decision process of whether I say one thing or another, right or left, or however you want to report that, then you might think, well, they should be exactly the same. I shouldn't see any differences in my results, but somehow timing does something different, even though the decision process is still just yes or no, or right or left.
Yes.
The report is exactly the same. So I find that really interesting, and I wondered, well, does that reflect that maybe more brain areas are involved in temporal decisions than, say, just V4 or something for color. That's one thing. And then the other thing I thought was, well, maybe, as people said before, you're always keeping track of time, and you maybe are always keeping alive the possibility of reproducing intervals with motor actions, whereas there's really no way to reproduce red. You know, you can say something about it. I guess you could paint it, but that's not a built-in kind of evolved capability. So I wondered if what this is reflecting is there's lots of brain areas online that are sort of getting behavior, you know, getting you ready to do timed behavior in the future, even though in this task you really don't have to do that. And so I wondered if a control experiment might be one in which knowing red or blue would involve different kinds of motor actions other than just pressing a button. I don't really know, you know, in other words, if you're seeing red or blue, and now what you have to do is something involved and motoric that requires a lot more than simply pressing a button. Maybe you'd see these differences in that kind of, in the EEG readout for the color task after the second stimulus finishes. So that maybe it's all just reflecting preparation to behave, even though you don't need to.
Yeah, I agree. So again, I think there's like between the color task and the time task, there's a bunch of differences, right? So as I said, one of them you can reproduce, you can do motor action, and with color that's not really the case. But I think another main difference which might explain is that this time you really need to integrate, right? So going back to what Jennifer mentioned in her first question, time is just one of those things that you can only make a decision towards the end. Which color, again, once you see it, you can already say, wow, this is more red or this is more blue. When you try to control for that, you kind of put time back in the game, right? So if you say, well, now, don't judge whether it's more red or more blue, judge whether on average it's more red or more blue. But then, again, you're kind of integrating color in time. Or I think what Fauci has been doing is similar to that with numbers, right? So you can kind of count numbers, but again, timing is coming back when people have to make this kind of judgment. So maybe this correlates that I'm saying, oh, look, this is correlated with time. It could just be correlated with any kind of temporal integration, which in our case is just people integrating duration. So I think it would be hard to get like a proper control of this. I understand. I see where you're getting. I think that's an interesting point, right? So maybe you could make so color, as you said, it's really like this is more red or this is more blue. But you could think about maybe people have to produce how red or how blue something is by pressing a button, like press a button for. It might involve time again, but something like, you see a color and you say, well, if you think this is really red, press a button for really long. If you think this is reddish, not that long, and the same thing for blue. And then people would have to, during the encoding, already kind of prepare whether they're going to press a button for long or not that long. So maybe that would, I wonder, that's kind of similar to what you're saying, right? Or
yes, and I mean, you're making me think as you speak of what if, to report this red or blue decision, or to rate it rather than to say yes or no, you had to do something pretty involved, like position a cursor in the red part of the screen rather than the blue. But it's actually hard to move it around, and you have to integrate all sorts of information about where on the screen the color really matches your judgment, I don't know, something like that, so that there's a lot of involvement of the motor system and the visual system, and then I wonder if in that case you might recover some of these effects, and then that would tell you something about how many brain areas or how much, something like that is involved in the decision, and then maybe all this temporal stuff is showing these temporal tasks are showing these effects because there's so much more involved in a way.
Yeah, well, but I think I would say that what Flatch is doing is really with numbers. I think that's the easiest thing to get closer to time. Really, like it's something that only increases in one direction. It can increase in different speeds. So I would. The first thought that comes into my mind is something in that sense. Because again, I think I mentioned before, right? So when you think, so in the beginning of the talk, one thing about color which I find interesting is that it can also dynamically change, but color can go back and forward, right? So it can start really blue, then it gets red, and then gets blue again, and that doesn't happen with time. Time just grows, and number has that, right? So numbers just grow, at least well, depending on the task. But so maybe that would be one way to kind of have a more a closer control to what you're saying.
I think Dr. Poole has another comment or question.
Yeah, it was just to, thanks. It was just to comment on this previous discussion just quickly. It was just to reply to Pat to say that in our original fMRI study using this time color paradigm, the color task was dynamic, and so subjects had to integrate color over the duration of the stimulus presentation. And what was really striking is that in the timing task, where we saw this whole network of different regions being activated, frontal, parietal, temporal areas, and so on, for the color task, despite the fact that it required an integration of information, we only saw activity in V4. So, there really seems to be something about time that is extra, aside from all of the integration type of stuff. And just as another quick comment, sorry, in terms of the discussion about what kind of stimuli you could use. So one paradigm that we've used with fMRI and that we're currently using with the EEG is a little dot that moves across the screen. And you have to judge either how long the stimulus lasted for or how far it traveled. And you can kind of manipulate both the distance and the duration of the stimulus. And so you can start to look at neural signatures of duration when you're paying attention to duration or when you're paying attention to some other feature like distance. And so, yeah, so I think that's quite a nice paradigm. It's similar to the idea that Andre was talking about with numbers.
Yeah, no, that's true. That's a perfect control because again, distance can only increase, right? So it changes dynamically and it can only get bigger. So in a way, that will control a lot of what Patrick's worried about.
Yeah, exactly.
All right, I think that's it. Thanks so much again for a great talk and a great discussion. I just wanted to end by telling that so our next virtual journal club will be on February 23rd, again at 10 a.m. Eastern. We'll have two speakers, Alison Hamlos and Fausto Forster, and we will share information on the talks pretty soon. So keep an eye out on the newsletter. So yeah, thanks everyone, and hope to see you all soon. Bye.
Bye-bye. Thank you.